Vaccines are a crucial tool for preventing infectious diseases, and they generally fall into two categories: traditional and rationally designed vaccines. Traditional vaccines rely on using whole pathogens, or significant parts of them, while rationally designed vaccines use modern scientific methods to target specific antigens based on detailed genetic, structural, and immunological insights. Traditional vaccines use entire pathogens, either in weakened or inactivated forms, or large parts of them to induce immunity by mimicking natural infection. Live attenuated vaccines. These contain weakened forms of the pathogen that are unable to cause disease in healthy individuals. Examples include the measles, mumps, rubella vaccine, and the oral polio vaccine. Inactivated vaccines. These vaccines use pathogens that have been killed or inactivated and thus cannot cause disease. An example is the inactivated polio vaccine. Subunit, recombinant, polysaccharide, and conjugate vaccines. These vaccines include only specific components of a pathogen, such as proteins or polysaccharides, to provoke an immune response. Examples include the hepatitis B and Haemophilus influenzae type B, or HIB, vaccines. While traditional vaccines have been highly effective for many diseases, their development can be time-intensive, making it challenging to quickly respond to rapidly mutating viruses like influenza or emerging pathogens. Rationally designed vaccines, often called next-generation vaccines, use modern scientific techniques to design vaccines in a more targeted and efficient manner. Instead of relying on whole pathogens or large fragments, these vaccines focus on specific antigens and epitopes informed by detailed data on the pathogen's structure and genetics. Reverse vaccinology. This approach starts with the pathogen's genome to identify genes that code for potential antigens. The selected proteins are produced in the lab and tested for their ability to elicit an immune response, a useful method for pathogens difficult to culture. For example, the meningococcal B vaccine, or Bexsero, was developed using reverse vaccinology. Structural vaccinology. This method uses imaging techniques like X-ray crystallography or cryo-electron microscopy to view the structure of key antigens at an atomic level. By visualizing these structures, researchers can modify antigens to enhance immune recognition. Epitope-based design. This approach targets only the specific parts, or epitopes, of an antigen that are most effective at triggering immune responses. This results in highly targeted immunity with a reduced likelihood of adverse reactions. For instance, epitope-based cancer vaccines are in clinical trials to prompt targeted immune responses against tumor cells. Computational and systems biology approaches. Computational models in systems biology analyze large datasets to determine which antigens might stimulate the strongest immune responses. This approach speeds up vaccine development by narrowing down candidates early. For example, COVID-19 vaccine development, including Pfizer-BioNTech and Moderna, used AI-based algorithms to identify spike protein candidates rapidly. Benefits of rationally designed vaccines. Precision and specificity. These vaccines target specific components of the pathogen, leading to stronger, more focused immune responses. Speed of development. Rationally designed vaccines rely on computational and molecular techniques, reducing the time needed for development and enabling rapid response during pandemics. Broad applicability. This approach is useful for pathogens that mutate quickly or are difficult to culture, making it versatile for both emerging and established diseases. Enhanced safety and efficacy. By focusing on selected antigens or epitopes, these vaccines may reduce the likelihood of adverse effects and improve both safety and efficacy. Examples of rationally designed vaccines. COVID-19 vaccines. The Pfizer-BioNTech and Moderna vaccines used mRNA technology to deliver instructions for making the spike protein, a crucial antigen of SARS-CoV-2, effectively prompting immune responses. RSV vaccines. Structural vaccinology helped stabilize the F protein of RSV, leading to more effective vaccines for RSV, a virus causing severe respiratory issues in infants and the elderly. Cancer vaccines. Epitope-based cancer vaccines, targeting specific tumor antigens, are being trialed to elicit immune responses that target cancer cells. In summary, while traditional vaccines have proven effective for many diseases, rationally designed vaccines offer a targeted, faster, and adaptable approach to vaccine development, particularly valuable for rapidly mutating pathogens and diseases requiring highly specific immune responses. Nature is ultimate.